We then moved into a different model. So raw 264.7 cells are just a macrophage line from mice that express mannose receptor. So, what we did for this one is we took in pink, we took BSA, so bovine serum albumin, that was monosylated. We labeled it with a fluorophore, and we added it to the cells and tracked fluorescence by flow cytometry, and that's this thick pink line up here. When we added mannan, a competitive inhibitor for mannose receptor, it reduced the rate of uptake, and that's the dotted line here for BSA. We then did the exact same experiment with our manganese peroxidase that was labeled with a fluorophore fluorescein, and we see in the solid line uptake into the cells, and then when we treat with competitor mannan, we see that that uptake rate is reduced. So this suggests strongly that manganese peroxidase is being taken up through mannose receptor, and that our planned delivery strategy is going to be effective. We then wanted to see how well this enzyme was tolerated in animals. So we did intravitreal injections, which is how the disease is treated currently. So we do injections into the eye with the enzyme. It diffuses in about 30 minutes to the retinal layer, and we see without treatment on the left, very nice morphology of the different neural layers and photoreceptor layers, and then an RPE layer here in the choroid beneath. When we treat with PBS, we see similar preservation of the macro structure, and even when we do 6 weekly treatments with our enzyme, we still see a strong assembly on the gross anatomy. We do see a little bit of morphological change here at the level of some of the cell bodies, and there's a bunch of things that we're doing to attenuate that I won't get into in this talk. But collectively, our first pass at a non-optimized recombinant enzyme seems to be really well tolerated in animals. So then we decided to move forward with our pivotal proof of concept, which were these 2 studies, where we took a genetic mouse model of the disease. So these are ABCA4 null mice. They accumulate A2E at a greatly accelerated rate. And we treated them with different concentrations of enzyme and looked to see if we were able to knock down in vivo the A2E. So the graph on the left is our first study where we treated with a low and a high dose, 24 micrograms and 112 micrograms. And then we did a second study where we treated with an intermediate dose. And when we pool those data together and graph it, we see a nice dose response in the combined graphs, suggesting that higher concentrations of enzyme are able to clear out the A2E. So, right now, we're in the process of submitting these findings for peer-reviewed publication. We're also in the process of fundraising to take this lead enzyme and do further development to turn it into a drug candidate that we can move towards clinical trials in humans. But we're really excited that this enzyme is capable of breaking down every major component of lipofuscin, every one that we've tested. It is able to be taken up into a cell through the desired pathway, mannose receptor endocytosis, and it appears that we can efficiently remove A2E from these animals in a concentration-dependent manner. And all of this was born out of a few things that we decided to tinker with inside of a living room when I was in medical school at Upstate. So with that, I thank you for your attention, and I'll take any questions that you guys have. Yes?
Did you ever finish medical school?
No. By the end of my second year in medical school, I'd raised over $1 million for the company, and it kind of became a, do I finish the MD, or do I go back and finish out the, or do I finish out the medical degree, or do I bet on the company? I ended up choosing to bet on the company, so I have an MBA, 2 years of medical school completed, and University of Miami's Miller School of Medicine actually has a program where I can do the didactic coursework online, but they allow the research that I'm doing at my company to satisfy thesis requirements. So the research that I presented here is going to be the basis of my dissertation in biochemistry through the University of Miami's Miller School of Medicine. Thank you. Other questions? Yes, sir.
More observation. It's amazing how people now change careers so often, and what you think you might be doing now is going to be so different from what you might be doing 5 years from now. It's just good to hear you hear and say those kind of things that you might be doing something you have no idea that even exists 5 years from now.
Yeah, that's absolutely the case. And, you know, it actually wasn't until my senior year in undergrad, I was actually a psych major that was going to go into Army intelligence, and somehow decided, I actually did a summer program at Fort Knox, saw a lot of people coming back from the war that were all blown up, and that kind of moved me in the direction of regenerative medicine and hence the stem cell company. So I completely agree with you. It's very unpredictable what directions you're going to move in. And the thing that we've kind of identified at our company is that if you put in strong processes and infrastructure and really smart people and surround yourself with a great team, those teams and those capabilities are very problem agnostic. So the very first slide I presented was kind of our circle of portfolio companies. Ichor Therapeutics is running probably 12 research programs for different organizations, as well as several intramural programs spanning stem cell research, gene therapy, enzyme therapy, and small molecule drug discovery. And the reason that we're able to do all of that is we put in very robust infrastructure where we can just be opportunistic about who we find that are good partners, people that have good ideas, and we can move very quickly on bringing in new clients, bringing in new partners, and kind of putting those projects through our pipeline. So as you guys move forward, you know, don't underestimate kind of these networks that you're building and the tangential skills that you're picking up. The thing that's so difficult is the life sciences these days is just very complicated. What I presented here spanned, you know, biochemistry and cell biology and medical stuff and husbandry. They're all multidisciplinary projects, so it's really important to get yourself immersed in a lot of different areas and be able to draw people from different disciplines together as you pursue these problems. And like you say, you have no idea where you're going to end up. Other questions? Yes?
It seems that you, with one of the scientific data, you found that a food substitute was for the loss of enzyme activity was manganese peroxidase. But you did have two other enzymes that were there that were less active as opposed to that one. Do those have any potential for that whole mannose pathway?
Yeah, so the question is, there were several enzymes identified. You chose to move forward with manganese peroxidase. What about the other enzymes? Yeah, so we did a lot of pilot work at the level of the wild type enzymes before choosing what to move forward with in a recombinant system. And there were a few things that influenced our selection of manganese peroxidase. First, it was by far the best performing enzyme in all of our cell-free, cell-based, and animal pilot studies. Secondly, and this is something that a lot of people don't appreciate, is manganese peroxidase is readily expressed in a recombinant system. The other two are much more difficult to express. And when we think about drug discovery and translational research initiatives, you know, we think about small molecule compounds that we take for a disease, but these are very easy to characterize because they're small molecules. You can figure out exactly what that compound is, so things like manufacturing and formulation are pretty straightforward, right? When we start moving into things that are more complicated, gene therapy, biologics like an enzyme therapy or even cell therapies, these are not homogeneous things. They're very diverse. There's a lot of variability in a batch-to-batch manner. So even though manufacturing might be the most boring topic that's ever poached the ears of a life science student, it's actually one of the most important issues when we're thinking about translating biologics. And the fact that manganese peroxidase outperformed, but also is readily expressible and is relatively easy to work with and stable, those things weighed in a lot in terms of guiding what we choose to move forward with. Excellent question. Going once? Yes, sir.
Another observation, my mother-in-law has macular degeneration. And I'm amazed that she can function as she can, with about 20 to 30% of the middle part of her eyes not being able to see anything, and all she sees is the outer 60, 70%, and still functions in a relatively normal way.
Yeah, the progression of macular degeneration is highly variable, and some people get hit really hard with it. Other people can have it stabilized, and they can, function pretty well. And this is one of the biggest things that I learned kind of moving through this whole process. We started off, I'm a cocky medical student, as we all are, with the startup company in my living room, and that was a super cool thing. But as the company began to progress, we started to have patients stopping by the lab. I started to get calls. Aaron, my COO, who's in the audience, we were in San Francisco in January to pitch an investment group about this very program. The day to go pitch, I got a call from a patient advocate, a mother whose son had been diagnosed with the juvenile onset form of this disease, which will quite plausibly render him blind by age 20 or so. And when you start getting those sorts of calls from patients, you realize it's not just a cool startup company anymore, but we truly believe that this program is one of, if not the best approach for treating this disease. And when you start having these patients call and you have those sorts of interactions, there's a certain sense of responsibility that takes hold where it's not abstract. You guys are out in your labs doing all kinds of cool projects, playing with C. elegans and worms and nematodes, all these other, flies, whatever. And it feels very, very unrelated, right? It's this project you're working on that's completely independent of your everyday life. When you're sitting in a translational research lab and you're at the intersection between basic science and discovery and moving that through a pipeline to create a product that can fundamentally change people's lives, it's very sobering, and frankly, it's something I wasn't really expecting as we went into this process. But as we as a company have matured, and I like to think I've matured a little bit as an executive, it's something that constantly reminds us to stay focused and to produce high quality products and high quality findings. Very good. Thank you very much for your attention.